Hello. This video is on cytokines, what we call them as soluble mediators, which are normally involved in cell-mediated immunity. Any soluble mediator produced by a cell, in general, we call them as cytokines. And if a soluble mediator specifically produced by macrophage or monocyte, we call them as monokine. For example, interleukin-1, which is a very important agent in causing the rising temperature, what we call as fever. Interleukin-1 is also called as an endogenous pyrogen. Pyrogen is an agent which causes fever, which is present within the macrophages. Whenever there is a pathogen entering into your body, probably the macrophages respond. Since because they are professional phagocyte, they actively produce, there is increase in concentration of this interleukin-1, which leads to the rise of temperature, what we call it as fever. And therefore, interleukin-1 is a very specific soluble mediator produced by monocytes, what we call them as monokines. In general, it is called a cytokine. Cyto refers to cell and kines refers to the soluble mediators. And if it is specifically produced by T lymphocytes and B lymphocytes, very specifically the immunocompetent cells, what we call them as lymphokines, then they are called as lymphokines. Lymphocytes producing soluble mediators, we call them as lymphokines. In general, the definition of a cytokine goes like this. Cytokines are very small, low molecular weight, secreted proteins which mediate or regulate immunity. They can also mediate and regulate the inflammatory process, the tissue damage, or they can also help in the blood cell production, what we call as hematopoiesis. Those proteins which are involved in the hematopoiesis, poiesis refers to generation, hemato refers to the blood cell generation, birth of the blood cells. And hematopoiesis is being induced by a protein called as hematopoietin. And these are produced in response to most of the immune stimulus. Cytokine in general, a term of a large group of molecules, which are also involved in cell signaling between the cells during an immune response. All cytokines which are involved in the cell signaling and which are involved in inflammation, regulation of immunity, and hematopoiesis are mostly proteins. Some are with sugar molecules. We call them as glycoproteins. They are synthesized and are released by white blood cells and tissue macrophages. But these cytokines are also proteins, peptides, or glycoproteins in nature, in general, what we say. And cytokines normally stimulate or suppress the functional activity of lymphocytes, monocytes, neutrophils, sometimes fibroblasts, and endothelial cells. These compounds modulate the function of other cell types, long known to be involved in the cellular immune response. That's why it's very predominantly spoken during the cell-mediated immunity. During humoral immunity, the main component which is involved in the destruction of an antigen is antibody molecule, which is produced by differentiated B lymphocytes. In cell-mediated immunity, the T cells, after getting sensitized, after recognizing an antigen, after binding to an antigen, after activation by an antigen, T cell, they don't involve themselves, they secrete these soluble mediators. And these soluble mediators will have different functions, and thereby they can bring about the elimination and eradication of this pathogen. So these products have additional effects that play an important role in both acute and chronic inflammation. There are 200 different types of human cytokines which have been identified so far. Cytokines are generated by mononuclear phagocytes, what we call them as macrophages, professional phagocytes. We call them as monokines, as I told you earlier. And if it is specifically produced by lymphocytes, we refer them as lymphokines. So the general properties when we speak about cytokines, cytokines are produced during innate immune response and also during acquired immune response. They are produced during immune and inflammatory responses. So inflammation is a process that takes place during innate immunity, and secretion of these mediators is transient and closely regulated. Cytokine effects are often redundant, and these proteins can influence the synthesis or action of other cytokines. Cytokines also mediate their effects by binding to specific receptors on target cells, and the expression of cytokine receptors can be regulated by a variety of exogenous and endogenous signals. So cytokine induce their effects in two ways. They can act in autocrine, that the same product of a cell can regulate the same cell. It's called autocrine. And if it is activating the adjacent cell, we call it as paracrine. And there is a third option, which is a very rare option, where cytokines can activate a far-off cell in a remote area. And if a cell produces cytokine, the cytokine can activate some other cell which is far distance from that cell that we call it as endocrine. So these are the three mechanisms by which they can activate. So there are the main sets of cytokines, which are the family of cytokines, which are grouped as interleukins as one important family. Second one is interferon, which is actually an antiviral protein. The third one is a tumor necrosis factor or the growth factor, which helps in the transformation of the cells, and colony stimulating factors. And there are certain molecules which can attract the cells to the site of inflammation, what we call them as chemokines.